Glucose is phosphorylated by ATP to form glucose 6-phosphate with the release of ADP by the action of an enzyme called kinase. That is first phosphorylation. Second is isomerization. You know the meaning of isomers, having same structural formula. That is glucose 6-phosphate is isomerized to fructose 6-phosphate by the action of isomerase. Glucose 6-phosphate to fructose 6-phosphate by the action of isomerase. And that fructose 6-phosphate now undergoes second phosphorylation. This reaction is second phosphorylation. Fructose 6-phosphate undergoes second phosphorylation. Here fructose 6-phosphate is converted into fructose 1,6-diphosphate by the addition of phosphate from ATP with the release of ADP. That is by the action of enzyme kinase. And this fructose 1,6-diphosphate now simultaneously undergoes the fructose 1,6-diphosphate now simultaneously undergoes cleavage as well as isomerase. Is cleaved into two trioses. Fructose 1,6-diphosphate is cleaved into two trioses, 3 carbon compounds. One is glyceraldehyde 3-phosphate, another one is dihydroxyacetone phosphate. Fructose 1,6-phosphate that is cleaved into two trioses, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. These are isomeric substances. Meant is DHAP is isomerized to G3P. DHAP in the sense dihydroxyacetone phosphate. G3P means glyceraldehyde 3-phosphate. They are isomers. That is by the action of isomerization by the enzyme isomerase. And now the second phase of glycolysis, that is oxidative phase. Here the reaction is this glyceraldehyde 3-phosphate. Let us take one compound out of these two isomers. Glyceraldehyde 3-phosphate undergoes phosphorylation and oxidative dehydrogenation. Phosphorylation means addition of phosphate, that is by means of H3PO4, and dehydrogenation means removal of hydrogen. Glyceraldehyde 3-phosphate is phosphorylated by phosphoric acid, that is H3PO4, and simultaneously it undergoes dehydrogenation or dehydrogenated to form 1,3-diphosphoglycerate. So after dehydrogenation, the pair of hydrogens are liberated, and those pair of hydrogens are accepted by NAD. NAD means nicotinamide adenine dinucleotide. They are accepted by nicotinamide adenine dinucleotide, and that reduces to NADH2, reduced nicotinamide adenine dinucleotide. And those pair of hydrogens are released from NADH2, enters or passes down to electron transport system to form 6-ATP. Here 2 hydrogen atoms were released, from 1 3-ATP molecules and from 2 6-ATP molecules. As a result of phosphorylation and oxidative dehydrogenation, 1,3-diphosphoglycerate is formed, and this 1,3-diphosphoglycerate undergoes first ATP generation here. So 1,3-diphosphoglycerate is converted into 3-phosphoglycerate, that is by the removal of a phosphate, and that phosphate is taken by ADP to form ATP under the action of enzyme kinase. 1,3-diphosphoglycerate is converted into 3-phosphoglycerate by the action of kinase, so that phosphate is released, taken by ADP to become ATP. And this 3-phosphoglycerate now undergoes isomerization. So 3-phosphoglycerate and 2-phosphoglycerate are isomeric substances or compounds. 3-phosphoglycerate is isomerized to 2-phosphoglycerate by shifting a phosphate from third carbon to second carbon, that is by the action of mutase. And this 2-phosphoglycerate now undergoes dehydration. Dehydration means that is by the removal of a water molecule, 2-phosphoglycerate is now converted into phosphophenol pyruvate. And that phosphophenol pyruvate that is formed by the action of enzyme enolase, this phosphophenol pyruvate undergoes second ATP generation to become pyruvate by the action of kinase. So that phosphate is released, that is taken by ADP to become ATP. So as a result, pyruvic acid or pyruvate is formed, which is a 3-carbon compound. Glucose 6 carbon, glucose 6 phosphate 6 carbon, fructose 6 phosphate 6 carbon, fructose 1,6 phosphate 6 carbon. 6 carbon compounds these are. And from here, all these are 3-carbon compounds which are formed in the form of 2 molecules. So that we have used 2 while the formation of ATP. So, in case of glycolysis, let us see the net gain of ATP, high energy bonded molecule, that is ATP. When glycolysis is completed, after the completion of glycolysis, in the sense when glucose is converted into pyruvate or pyruvic acid, 2 molecules of pyruvate or pyruvic acid is formed, that is by oxidation of 1 molecule of glucose. Here 2 ATP molecules are used at initial stages. 2 ATP molecules are used at initial stages and 4 ATP molecules are generated at subsequent stages. Here is 2 and here is 2. 4 ATP molecules are generated at subsequent stages. So net gain of ATP after the completion of glycolysis is 4 minus 2 is 2 ATP molecules. In addition, some oxidation also takes place, that is removal of electrons and hydrogen ions. Hydrogen ions and electrons bounded to NAD here, that is NAD to NADH2. So 2 NADH2 are formed by the oxidation of 1 molecule of glucose. Later, hydrogen ions and electrons are used in electron transport system. So final product of glycolysis is 1 molecule of glucose is oxidized to 2 molecules of pyruvate and 2 molecules of NADH2 and 2 molecules of ATP. So after the formation of pyruvate or pyruvic acid, this enters into Krebs cycle with an intermediate step, that is formation of acetyl coenzyme A, or that is in the presence of oxygen, Krebs cycle. And in the absence of oxygen, this pyruvate undergoes fermentation. Either lactic acid is formed or ethyl alcohol is formed. This is about lactic acid formation is called lactic acid fermentation. Ethyl alcohol formation is called alcoholic fermentation. So this is about the steps involved in glycolysis. I think you will understand the steps involved in glycolysis process, which will going to be asked for 10 marks in your examination. If you have any doubts after listening to this video and also after going through the notes that I am going to give you, you may ask your doubts or any queries without any hesitation. Thank you.